All right, hello. Today we are going to be covering chapter 17, which is the digestive system. Here are some roots, prefixes, and suffixes that you will need to know as we go through this chapter. Some of them, of course, you already know. Hopefully, whoops. Let's see. And, gastro means stomach, hepat, liver, peri means around. We've talked about microvilli before. All right, so, okay. So digestion. Basically, we can break digestion down into two different categories, mechanical digestion and chemical digestion. So you eat your Big Mac, and mechanical digestion is just taking that Big Mac and chewing up the food. So you're breaking the huge Big Mac into smaller particles. We're not actually breaking those particles down, and we're not taking carbohydrates and breaking down into the sugars. We're not breaking down the fat. We're not breaking down the proteins. If there is any protein in Big Mac, I'm not sure yet. But so mechanical digestion is just the process of using your teeth to break down large pieces of food into smaller ones. We're not changing the chemical composition. You're mixing your food with saliva. Chemical digestion is when we actually start to break down those smaller food particles. And chemical digestion actually does start in the mouth. Chemical digestion, we know that proteins are going to break down to form amino acids. Lipids are going to break down to form amino acids. Carbohydrates are eventually going to carbohydrates are going to eventually break down to disaccharides and then monosaccharides, and eventually, hopefully, we'll use that glucose. Those monosaccharides will get converted to glucose to use for energy, and then we can extract the vitamins, minerals, and water from the food that we take in. So we have an enzyme. I said chemical digestion begins in the mouth. We actually have an enzyme called salivary amylase that will start breaking down carbohydrates into disaccharides. So let's say you take a piece of white bread, you put a piece of bread in your mouth, and you don't chew it. You just let it sit in your mouth, and after about 5 minutes, that saliva will have begun to break down those carbohydrates to disaccharides, and you'll start to taste a sweet sensation in your mouth because you're actually tasting disaccharides. So salivary amylase is an enzyme that will catalyze carbohydrates to form disaccharides. And so chemical digestion does begin in the mouth. All right, so organs of the digestive system, we know, will carry out the process of mechanical and chemical digestion. So when we take in food, we call that ingestion. And when we poop out whatever we poop out, that's called defecation. When we move stuff around in our digestive system, we swallow the food and our stomach mixes it up and it moves it through our small intestine, our large intestine. That's called propulsion. And eventually we'll break down our Big Mac into proteins. We'll break it down, and we'll break down those proteins, carbs, and fats, and vitamins, and minerals, and water, and we'll absorb those nutrients. However many nutrients are in a Big Mac, again, is questionable. So the digestive system consists of this thing called an alimentary canal and accessory organs. So the alimentary canal we'll see on the next slide, and we'll talk about the accessory organs later on in the chapter. So basically the function of the digestive system is to take in food or to ingest food, to break down the food, to absorb those nutrients and to defecate any unabsorbed material. So the alimentary canal, remember I said the digestive system made up of two parts, the alimentary canal and the accessory organs. The alimentary canal is basically the passageway that the food goes through from the time that you ingest your Big Mac or Taco Bell. You take in your food and it goes into your mouth. And then it's going to go into your, like the back of your throat called your pharynx, and then into your esophagus, and then down into your stomach, small intestine, large intestine, anal canal, and then you poop out or you defecate the unabsorbed material. It's a muscular tube. It's about 8 meters long. The majority of that is going to be obviously your small intestine. And then we have the accessory organs, and that's going to be kind of what helps break down that that chemical digestion. So like your salivary glands, I talked about salivary amylase, your liver, gallbladder, and pancreas, and we'll talk about all of those throughout this chapter. All right, so when we look at the alimentary canal wall, it's going to be composed of 4 layers, and we'll talk about these layers from the innermost to the outermost. And you're going to have to know them from the innermost to the outermost, or the outermost to the innermost. So figure out a way to remember them. So the innermost layer is a mucosal layer. It has a membrane. All it is really is it's a folded area that is going to increase the surface area of the membrane. So here you can see that the mucosal layer, and it has these little projections called villi on them. And as if we're talking about the small intestine, as food particles move over these microvilli, these food particles are going to kind of wiggle their way down into these tiny little spaces, and that's going to increase the absorption area, the absorptive area, or increase the surface area so that more nutrients can be extracted from whatever we've just eaten. And then we'll see that there's some mucus that'll be secreted by these things called goblet cells. And then we have these enzymes that are going to have to be secreted in order to break down some of the food. And that next layer is called the submucosal layer, and it's basically just a loose connective tissue layer. It has some blood vessels. You can see some lymphatic capillaries in there, some lymphatic vessels. There's some nerves in there. And basically it's just going to nourish the mucosal layer, and it's going to help transport those food particles that were just absorbed. All right. So then our next layer, it's called a muscularis layer. As the name implies, it's a muscular layer. It's also called the external or externa layer. It's a double layer. You can see here the muscularis layer. And since we're talking about our visceral organs, it's going to be a smooth muscle layer. It's going to be composed of circular and longitudinal layers. So remember that in the esophagus and the small intestine, we have this thing called peristalsis, and it's just like this wave-like or rhythmic-like contraction that's going to help move food through the alimentary canal. And then the outermost layer is a serosa layer right there, and basically that is just there to help with prevent or eliminate any friction. Remember that we have that visceral layer and the parietal layer. We talked about that in AMP1, seems like 14 years ago now. So remember the visceral layer is the inner layer, that's the one that's going to be right next to the organ itself. And then the parietal layer is going to be the layer that is lining the wall, like in this case, the abdominal wall. All right, So here you can just see kind of like a summary slide of the 4 different layers. The inner layer is going to be the mucosal layer, then the submucosal layer, and then the muscularis layer, and then that outer layer is going to be the serosa layer. All right, and we already talked about the peristalsis a little bit, but the movement of food particles in the alimentary canal consists of 2 types. We have a mixing movement and a propelling movement. So with mixing movements, we need to be able to mix our food with the gastric enzymes and all that stomach acid and all the secretions that come into our stomach so that those food particles can be broken down. It doesn't necessarily move the food particles in one direction, but basically, it's just the food is going to be in our stomach to kind of mix up and kind of really work those digestive enzymes into the food particles that are sitting in our stomach. So remember, mixing movements, they don't move the food in any one direction. And so we'll see this we'll see the mixing in the stomach as well as the small intestine. In the small intestine, what we do, when we see this is, it's called segmentation. And, when we have food in the stomach, what will happen is you eat, your Big Mac and fries. That Big Mac and fries, once it's mixed in the stomach, your stomach doesn't open up and say, okay, boom, let's throw all of our Big Mac and fries into the small intestine. It will kind of release a little bit at a time. And then the small intestine will do what it needs to do, and then the stomach will release a little bit more. The small intestine will do what it needs to do. Small intestine, and the cycle will repeat itself. And you can see over here in that part B shows this segmented movement of digestive material through the small intestine. So in this first step, the digestive material comes in the small intestine, and then in this tube with the digestive materials kind of shrinks, making the material move forward. And then in that third step, due to shrinking of the tube, the digestive material has moved kind of on both sides. And so it kind of helps mix the food still with the gastric secretions. And then the propelling movement is going to help obviously propel the food particles forward through the alimentary canal. And we talked about peristalsis, moving the contents through the small intestine. That was the example I used. But peristalsis actually occurs from the pharynx through to the anus. So the pharynx, remember, is in the back of your throat, and then once you swallow your food, your esophagus does peristalsis, your stomach, your small intestine, your large intestine, all of those places do peristalsis. And that's going to be accomplished by these rings of contraction. So in this case, the smooth muscle, if we go back and look at that muscularis wall, that smooth muscle, remember I said it's in circular and longitudinal groups. So rings of contraction, that would be those circular muscles are going to help move the tube kind of forward via peristalsis. All right, so when we talked about, I think it was in chapter 10, 11, maybe 11, I'm not sure. I think chapter 11, I said we talked about the parasympathetic nervous system and the sympathetic nervous system. And I said that every organ system has both sympathetic and parasympathetic innervation to it. So the digestive system does have parasympathetic and sympathetic nervous system innervation. And I also said that usually one system will be primary, parasympathetic or sympathetic, and one system will be secondary. And we'll see that in the digestive system that the same rule applies here. And the submucosal plexus is what's going to control the secretions, and this myenteric plexus is going to be what controls the gastrointestinal motility, or moving food particles through the gastrointestinal or GI tract. Now, remember that the autonomic or automatic nervous system is in control, because these are processes that we do not have any control over. This is our involuntary nervous system, or the autonomic nervous system. And so think about what happens when you encounter the bear in the middle of the woods. Your body really doesn't care about your digestive system when it comes to fight or flight. And remember, we now should say fight, flight, freeze, or fawn. So because your body, and if you fight or flight, or freeze or fawn, that's going to be your sympathetic nervous system. And remember, your parasympathetic nervous system is your rest or digest. So here, your parasympathetic nervous system is going to be primary for the digestive system, and your sympathetic nervous system is going to be secondary. So your parasympathetic, if you have stimulation by the parasympathetic nervous system, that's going to increase any activity of the digestive system. So you're going to see increased enzyme secretion, increased motility. You're going to see increased salivation. And if you remember from chapter 11, the vagus nerve, or your cranial nerve number 10, is going to be in control, is going to control a lot of what happens with your digestive system. So on the flip side, your sympathetic nervous system is going to inhibit digestive actions, or actions of the digestive system. So sympathetic impulses arriving from your sympathetic nervous system is going to decrease any enzyme activity or and motility. So in the, you encounter the bear in the middle of the woods, your sympathetic nervous system is going to kick in. So pretty much your digestive system is going to shut down, because your body's more worried about survival and either fighting the bear, running away from the bear, and so your body at this point doesn't care what's happening with your digestive system, so it's just going to kind of shut it off. We also have these things called enteroendocrine cells. Entero just means gut. So basically we have these endocrine cells that are found in your gut. And in this case, gut, we're going to mean as stomach and small intestines. And their purpose is just to secrete hormones that are going to regulate your organs and the processes of your gastrointestinal tract. So one of the accessory organs that we're going to first talk about is the mouth. It's going to be obviously the first part of the alimentary canal. That's where we take in food. It includes your oral cavity. We also call it the buccal cavity. And we have this thing called the vestibule. That is kind of like underneath your bottom of the lip. So we also have, the lips, the cheek, the tongue, the palate. We have two palates. There's the hard palate that's going to be on the roof of your mouth, just posterior to your teeth. And then we also have a soft palate that's going to be on the roof of your mouth, just posterior to the hard palate. And then we have that little hanging down thing called the uvula. So right here is your uvula. So there's your or your hard palate, and then your soft palate's going to be here, and then the little hanging down thing is called a uvula. And we know that we take in food, or we ingest food into our mouth, and we start chewing food. I'll move this. We start chewing food, it's called mastication. Mastication is just the process of chewing food. So we start this mechanical breakdown or mastication of solid food particles, and we're gonna make the big food particles into smaller food particles. Mechanical breakdown, and we're gonna mix those food particles and with saliva. And once we mix those food particles with saliva, we now say that this is a bolus. So food particles plus saliva is a bolus. So bolus equals food particles plus saliva. And then we also know that the mouth is going to function as an organ of speech. And then we also have sensory reception. We learned in chapter 12 that our mouth or our tongue acts as a taste receptor. All right, your cheeks. We all know the cheeks form the lateral walls of the mouth, and we have muscles that allow us to chew. And your masseter muscle. So when you bite down those muscles right here, your masseter muscle is actually the largest or the strongest muscle in your body. Most people would assume it would be like your one of the muscles in your quads or your quadriceps. It's actually your masseter muscle. Muscles in your cheeks also help us with facial expressions. Your inner lining of your cheeks form this stratified squamous epithelium, which helps keep the inner lining of your mouth moist. And the reason why we have this stratified squamous epithelium and not a simple squamous epithelial layer is to help with not to help with friction, but because of friction. When we bring in food particles, and as we're chewing, that chewing process and the food rubbing up against our cheeks does create friction. It's going to wipe off some of those outer layers of the stratum corneum, and if we only had a simple layer, one layer, pretty much after one ingestion of one meal, you would not have any cheeks left. So that's why we have a stratified layer, not a simple layer. And then we have lips, highly mobile structures surround the mouth opening, and that's going to allow us to judge temperature, texture of food. All right, so the tongue itself. So the tongue, we can call it a lingula. We have the tongue is actually basically a muscle. It's very thick. It's an intrinsic and extrinsic organ. It's going to occupy the floor of the mouth. And when the mouth is closed, your tongue pretty much fills up your entire mouth. We have this little thing called the lingual frenulum. If you lift up your tongue, that's a little thing that connects the tongue to the floor of your mouth. So that little structure that connects your tongue to the floor of your mouth is called a lingual frenulum. And there's a condition out there called ankyloglossia. And that's where your lingual frenulum is kind of really shortened so that your tongue is pretty much anchored to the floor of your mouth. And as a result, you're going to have difficulty talking or difficulty with speech and manipulating food. And then we also have these things called papillae. And you can see the little papillae here on your mouth. And that's what is going to contain your taste buds. And then we also have these things called lingual tonsils. If you can stick out your tongue far enough, you can actually see the lingual tonsils on the back of your throat. All they are just lymphatic tissues, lymphatic tissue masses on the back part or the root of the tongue to help with immunity. All right, so this palate forms the roof of the oral cavity. We already talked about the hard palate and the soft palate. The hard palate's going to be, consist of this palatine process of the maxilla and the palatine bones. So here you can see, here's where your hard palate is, there's where your soft palate is, and then there's the uvula, that little hanging down thing. It's just a projection of the soft palate. All right, and your uvula, all it is, I said, it's a projection of this from the soft palate, but it's made up of like connective tissue and some glands and some small muscle fibers. But it does secrete large amounts of saliva that help keep your throat moist and lubricated. But it also helps keep fluids and food from ending up in the space behind your nose when you swallow. So it does serve some sort of purpose. We also have these things called palatine tonsils. You can see those here. Those are the tonsils that you see when you look in the back of your throat. They're again just lymphatic masses to help provide immunity, and if they are swollen, they can interfere with your breathing and swallowing. I know when I had my tonsils removed, it was because they were so overgrown. I was actually snoring because of it, and I had trouble swallowing food. I had to chew my food into really, really small bites before I could swallow it. Not that you needed to know that, but oh well. All right, pharyngeal tonsils, we also call those adenoids. So typically if you have your tonsils out, we also remove your adenoids. So you might go in for a tonsillectomy and an adenoidectomy. Again, they're just more masses of lymphatic tissue in the posterior wall of your pharynx. So they're located on the backside of your throat. All right, so teeth. Teeth is pretty much is the hardest structure in your body. We don't classify teeth as part of the skeletal system because they're not bone. And we use our teeth for grinding up food or breaking up food into smaller pieces. We call them baby teeth and adult teeth, but what we should be calling them are either primary teeth, or baby teeth, and deciduous teeth, or secondary teeth, which are adult teeth, or permanent teeth. And we have 20 primary deciduous or baby teeth, and we have 32 secondary permanent or adult teeth. And they consist of, central incisors, lateral incisors, canines, which are your cuspids. First premolars are also called your bicuspids, molars, and then your wisdom teeth. Anyway, so your teeth, as we said, to break down food into smaller pieces, that's also mechanical digestion, remember. And it also mixes food with those enzymes. Remember that salivary amylase that's going to start the chemical digestion process. Incisors are going to be used for biting. Canines can be used for grasping and tearing. Molars and premolars are going to be used for binding. And I'll just put that there. All right. Now, your teeth, again, we have this thing called crown, root, and that's going to meet at the neck of the tooth. And we have the enamel that's going to cover the crown of the tooth. And dentin is going to be what makes up the majority of the tooth, and it's going to surround this pulp cavity. And we have this thing called a periodontal ligament that's going to hold this root in place along with cementum. And I'll show you this first. So here you can see the crown of the tooth, and then the enamel is what's on the outside. And then we have this dentin in yellow. And then we have our blood vessels and nerves. And the cementum thing, this cementum is right here. Here's your gum line, and this is your alveolar process or your bone. So this whole thing that we can't see would be called the root. And if you go to the dentist to have a root canal, basically what they're going to do is they're going to be drilling all the way down into here and kind of killing off this nerve that's down here. So just remember, the root is where your blood vessels and your nerves are going to be residing. All right, so I said that we have oops, you really can't see that, red on red. We have about 20 primary teeth and we have 32 secondary teeth. Oh, totals are down here. All right, you don't need to know this chart, but here's just what when people start to get their adult teeth or their secondary teeth or their permanent teeth, and just kind of what type of teeth do what. All right, dental caries, also known as cavities. So when we talk about cavities, we are talking actually about the enamel. So the white portion of the tooth up here. And essentially it is you eat too many sticky foods or sugary foods or too many carbohydrate containing foods. Remember that carbohydrates break down into disaccharides and into monosaccharides. And bacteria thrive on sugary material. So bacteria on the teeth actually metabolize these carbohydrates and they produce these acidic byproducts that are going to damage the enamel and the dentin and actually destroy it. And if you need your words of wisdom from the American Dental Association, brush and floss twice a day. Go to the dentist twice a year, maybe do some fluoride treatments and some sealants. All right, summary slide of what does what in our mouth. All right, so salivary glands. We have several different salivary glands in our mouth. Basically, salivary glands are going to secrete saliva. Which is going to help moisten food particles and to help kind of keep them bound together. And it's also one of the other functions is to dissolve food so that it can be tasted. Remember in chapter 12, we learned that, when you smell chemicals, we have that mucosa or that kind of snot type of thing that will dissolve the odorants so that you can smell them. So here, saliva is going to dissolve the food particles so that we can taste them. And salivary glands are going to contain two types of secretory cells. They can either have serous cells or mucosal cells or mucus cells. Mucus cells, all they are to secrete mucus. And that's just going to bind food particles. Remember, when we have food plus saliva is going to equal a bolus. So mucus cells really only act as a lubricant for swallowing. Now, serous cells, on the other hand, are going to contain certain enzymes and bicarbonate ions. And I'll just write, bicarbonate here. Bicarbonate is just HCO3. And so we've already talked about salivary amylase. That's going to begin that chemical digestion of carbohydrates. It's going to split, the that carbohydrate into disaccharides. So it's going to split the starch into glycogen and starch, glycogen, whatever, into disaccharides. And if you remember from what was it, chapter 2 or something like that, that chemistry chapter, it's a hydrolysis reaction. It is an anabolic reaction. Remember, anabolic reactions break things down. Bicarbonate ions are going to help keep that pH between 6.5 and 7.5, because remember, bacteria love acidic environments. So as I'm sitting here drinking my coffee, if I didn't brush my teeth, bacteria would like that acidic environment because coffee's acidic. But bicarbonate ions that are released from these serous cells, my saliva, are actually going to help protect my teeth. And I'll just mention here that I think, everyone knows that your salivary glands can be stimulated not necessarily by food entering your mouth, but just by thinking about food or by looking at pictures. So if you're looking at a magazine, or if you're looking at a TV commercial or something like that, and you see a picture of food that looks enjoyable, your body might start to secrete saliva. All right, so salivary glands themselves, we actually have 3 major pairs of salivary glands. We have these things called parotid glands. Parotid glands are going to be found more on the side of your face, kind of more right in front of your ear. If you do the lemon, put lemon or taste lemon, and you get that little sharp feeling, that's going to be your parotid glands. Parotid glands are the largest of your salivary glands. And that's where about 25% of your saliva is going to come from. We also have these things called submandibular glands. Submandibular glands are on the floor of the mouth. You can see here submandibular glands. They're on the floor. Oops, wrong one. Yeah, submandibular glands, floor of the mouth. They're going to secrete both. They have serous cells and mucus cells. So they're going to secrete the mucus and saliva. So they're going to contain about 70% of the saliva that is secreted in our mouth. And then we also have sublingual glands. Sublingual is just mean under, sub is under, and lingual is tongue. So under the tongue, sublingual glands. And those are going to be mainly mucus secreting cells. And even though I said mainly mucus, it does secrete about 5% of the saliva that's in our mouth. And we have these minor glands that are going to be scattered throughout our tongue, palate, cheeks. I mentioned the uvula before, and just going to help keep the lining of our mouth moist. We have these things called von Ebner's glands that are found on the tip of the tongue. Basically, these release these things called lingual lipase. And lingual lipase, remember, usually ASE means to break down. Always doesn't mean always. So in this case, triglycerides are going to get broken down into free fatty acids, diglycerides, and monoglycerides. And here it says it releases less than 1% of our 1% of our saliva. All right, so here you can see just a histology slide of the serous cells, some mucosal cells. Here's a little duct. If we're talking about a duct, remember that's an exocrine function, not an endocrine function. Remember, endocrine releases into the bloodstream, exocrine releases to a free apical surface, such as a duct. There's a duct down here. All right. Summary slide of parotid, submandibular, and sublingual. For the exam, you're going to have to know where each of the glands are in layman's terms, on the side of the mouth, under the mandible, under the tongue. You're also going to need to know that they're going to produce and secrete saliva. But I will say, read the test question very carefully. Obviously, read all test questions very carefully, but it will try and the test question and the answers are going to try and trick you a little bit. So it'll say, the parotid, it'll say parotid glands on the floor of the mouth, or it'll say submandibular glands posterior to and not anterior to, or it will say sublingual glands in the floor of the mouth anterior to the tongue. So just make sure you know anatomically where they are and read the test questions so that you don't mix up, inferior or superior, something like that. All right, so your pharynx and your esophagus. Remember, your pharynx is kind of like your throat, and the esophagus, both the pharynx and your esophagus, they do not do any type of mechanical digestion. They don't do any type of chemical digestion. All they are just food passageways that transport food from one area to the other. So from your mouth to your stomach, food has to go through your pharynx and your esophagus. All they are just muscle walls, muscular walls. So your pharynx is going to be posterior to your mouth, and it's going to extend from the nasal cavity to your esophagus. We can divide it up into 3 different places. We have our nasopharynx, oropharynx, and laryngopharynx. So your nasopharynx is posterior to the nasal cavity. And typically it's just an air passage. And that's going to be where the openings are for your auditory tubes. If you remember back from chapter 12, we talked about that the auditory tube. Your oropharynx, oro means mouth or oral cavity, so it's posterior to your oral cavity. It's an air and food passage. So in this case, your pharynx, food would go through your oropharynx, and laryngopharynx is posterior to the larynx and a passageway to the esophagus. So food would pass through the laryngopharynx. So of the 3 parts of the pharynx, the food would pass through 2 out of the 3 parts of the pharynx, the oropharynx and the laryngopharynx. And the esophagus is a tubular organ that's going to connect the pharynx to the stomach, and like I said, it's just a food passageway. So swallowing can be divided into three stages. The first stage, second stage, and third stage. So that first stage is a voluntary stage. So we have, we're chewing our food and we're creating that bolus by mixing the food with the saliva. And our tongue is going to push that food back towards the pharynx, or more specifically, the oropharynx. And then the second stage, which is an involuntary stage. I'll do that too. Um, mm, um, that's going to begin once the food reaches the oral pharynx, and once the food reaches the oral pharynx, it's going to trigger this swallowing reflex. You know, we're going to stimulate these receptors, and it's going to trigger this swallowing reflex, and you're going to have to know this this swallowing reflex. So basically, your tongue is going to press up against the uvula and the soft palate, and that's going to cause the uvula and soft palate to rise, and it's going to close off the nasopharynx so that food can't go from your mouth to the back of your throat up into your nasal cavity. Um, and then your hyoid bone and your larynx are going to elevate or rise up. Your epiglottis, which is attached to the larynx, there's your epiglottis there. You can kind of see it right there. Your epiglottis is actually going to close off the top part of the trachea so that when you do swallow the food, your food is not going into your trachea. It's going to go into your esophagus. So then your longitudinal muscles of the pharynx are going to contract toward the food, and then your constrictor muscles are going to relax so that the esophagus opens up, and then peristalsis is going to work to force food into the esophagus. And then that third stage is also an involuntary stage, and that the peristaltic activity, or the peristalsis of the esophageal walls, is going to move that bolus from the pharynx to the stomach. So remember that second and third stage is an involuntary reflex, or an involuntary stage. So this is actually why we can swallow food when we're upside down, is because of the second and third stage. So if you look at what happens physiologically during that second and third stage, you know, the tongue pressing up against the uvula to close off the nasopharynx. The epiglottis closing off to protect the trachea. The peristaltic waves moving the food into the esophagus, and then the third stage down into the stomach. That's going to work whether or not we're right side up, laying down, upside down, or whatever. So just remember that this allows us to be able to swallow food while we're upside down. All right, so the esophagus is just basically a muscular passageway that food travels through. It goes from the pharynx to the stomach. It's about 25 centimeters long. Let's do the math on that. 25. So it's about 9.84 inches. So, 9.84 inches. There we go. All right, and oh, my backpack just fell, and it scared the living daylights out of me. Oh my gosh. All right. So it penetrates the diaphragm through this thing called an esophageal hiatus. Oh, I'm on the wrong slide for you guys. Sorry about that. All right. Penetrates the diaphragm through the esophageal hiatus. So right here is where the esophageal hiatus is. So the esophagus comes down, and it's going to enter into the stomach through, here's your diaphragm muscle. And there's a little opening in your diaphragm where the esophagus actually kind of goes through. And if you remember, from actually the slide that I was on up here, that we had those four layers from inner to outer, is the mucosa layer, the submucosal, muscularis, and serosa layer. We'll see the same thing here. And there we go. So um, in the submucosal layer, we see that there's these mucus glands, which are going to moisten and lubricate the lining of the inner wall with mucus. And that's going to help reduce friction. And we see that there's also a stratified layer, not a simple layer. We like the stratified layer to because as we swallow that bolus and as the bolus moves down the esophagus, it's going to kind of tear off or slough off some of those outer layers of the stratum corneum. So we want a stratified squamous epithelium so that we don't tear open our esophagus when we swallow food. In the muscularis layer, remember that's our third layer from inner to outer, we can see that it's made up of skeletal muscle, skeletal and smooth muscle, and smooth muscle. And then in our, and I said that the esophagus enters the stomach through the esophageal hiatus, which is like the opening in the diaphragm. Where the esophagus connects to the stomach, there's a circular ring of muscle called a cardiac sphincter or a lower esophageal sphincter. It has two names, and it's called the cardiac sphincter because it's near the heart, even though it has nothing to do with the heart. We're talking about the digestive system. So typically we'll just call it the lower esophageal sphincter, and that's going to help regulate food passage into the stomach, but it also, more importantly, prevents food from going backwards. So we don't want food going from the stomach back up into the stomach or into the esophagus, or also we don't want that stomach acid to go from the stomach, or the gastric enzymes. We don't want those to go from the stomach to the esophagus. All right, the stomach itself is it's a J-shaped pouch like organ. So here I guess we can kind of see it's a little J-shaped. To me it's more kidney bean shaped, but we'll say J. 25 to 30 centimeters long, so about 10 inches long. It's about the same length, a little bit longer than your esophagus. It's just inferior or below the diaphragm, and it's in the upper left portion of the abdominal cavity. And it has these things called rugae, and all rugae are just folds. You can see the rugae here. The rugae are just kind of like folds in the stomach wall that allow the stomach to expand. Because the stomach itself can hold about 4 liters worth of whatever, food, enzymes, liquid, combination thereof. And we know that the stomach is going to receive food from the esophagus. And then we'll say removes food, bolus from the esophagus, and then it's going to mix that bolus. We'll just do this because it's not food anymore. So it receives the bolus from the esophagus, and it's going to mix that bolus with gastric juice. Now, once we mix that bolus with gastric juice, we now call it chyme, C-H-Y-M-E, chyme. So chyme is food plus saliva plus gastric juice. And we see that the stomach doesn't really have a lot of absorption properties, but or that the stomach doesn't do a lot of digestion, um, with the exception of proteins. Some more, proteins are broken down into amino acids, and so this will start in the stomach. And then we know that food will go from, I guess now I should say, chyme will move from the stomach into the small intestine. And the stomach has the stomach will have 2 layers, sometimes 3 layers of muscle. And remember that because we're talking about our visceral organs, it's going to have smooth muscle. We have an outer layer. It's a longitude. It's the muscles are laid. The arrangement of muscle is going to be laid longitudinally. So here you can see the longitudinal aspect of the muscle, and we also have an inner layer, which is a circular layer. So you can see the circularness of the circular pattern of the stomach wall. And then it says some portions have a third of oblique muscle fibers, and oblique just kind of means to run at an angle, or at an angle. And we can see that there's 4 parts of the stomach. We have this thing called a cardia, a fundus, a body, and a pylorus. So your cardia, okay, so here is your lower esophageal sphincter, or your cardiac sphincter, and then right after that is your cardia. And then your fundus is the top portion of your stomach. And then we have the body of your stomach. That's going to be the main portion of your stomach. And then we have another sphincter to help prevent backflow and also to help regulate forward flow. We have this pyloric sphincter to help regulate flow from the stomach to the small intestine, and also to prevent backflow from the small intestine back into the stomach. So we have this pylorus area of the stomach as well. So cardia, fundus, body, and pylorus. And then we also have 2 curvatures of the stomach. The greater curvature, which is the big curve, and the lesser curvature, which is the smaller curve. Oh, and it looks like I jumped ahead of myself. So we've already talked about the cardia, is where the lower esophageal sphincter is. The fundus is the top part of the stomach. This is where a lot of your air is going to be at, just because, if you have a bunch of liquid and air is going to be above the liquid. So if I said that your stomach can hold 4 liters. So if you drink a liter of water, your water is going to be down here, and then all this is going to be air. Your body is going to be the main portion of your stomach. It's going to lie between the fundus and the pylorus. Your pylorus itself is going to be the last part of your stomach. We have that pyloric sphincter. And we have that pylorus can be divided into 2 different areas. Your pyloric antrum. Think of antrum as like a waiting room. So we have our pyloric sphincter, and then our pylorus is the whole section. So our antrum, it would be like the waiting room. And then our canal is basically a narrowed version of the waiting room. And we have this condition out there, typically seen in like Newborns is not common in newborns, but we typically, when we do see it, we see it in newborns, called hypertrophic pyloric stenosis. So we're talking about the hypertrophic pyloric stenosis, basically an overgrowth of muscle tissue that is blocking the passageway from the stomach to the small intestine. So we're talking about the pyloric sphincter overgrowth. And it usually leads to frequent vomiting. So if you're going to be working in the peds unit, if you're going to nursing school, just kind of keep that in the back of your mind. All right, so secretions, gastric secretions. We have these tubular gastric secretions that are going to indent inward from these gastric pits. All right, so gastric pits. You can see the gastric pits are these long, essentially abysses. I don't even know what the plural of abyss is. Is it abyss? Abysses? All right, we'll say it's abysses. So these gastric pits, we see these villi, these extensions that are going to come out from the gastric pits, and we're going to see that they have 4 types of secretory cells: mucous neck cells, chief cells, parietal cells, and enteroendocrine cells. And collectively, they're going to produce this thing called gastric juice. So here you can see the parietal cells. You can see chief cells. You can see the enteroendocrine cells. And what was the 4th one? Mucus cells. Mucus cells, remember the mucous neck cells, there is some right there. All right. Right, old cheek. All right, so and here it just says basically it says this structure over here. So basically what this means is that these chief cells are going to secrete pepsinogen. And I'm just going to kind of type this in the notes as I say this. Chief cells are going to secrete pepsinogen. Parietal cells secrete hydrochloric acid. Now, you see this ogen, O-G-E-N suffix. Usually anything that is ogen is an inactive form of whatever enzyme we're talking about. So pepsinogen is the inactive form of pepsin. Trypsinogen is the inactive form of trypsin. We can say, let's say we have an enzyme called James. So James ogen would be the inactive form of James. Or take your name. So nameogen is the inactive form of name. And we have these catalysts or enzymes that are going to convert the inactive form to the active form. So hydrochloric acid converts pepsinogen to pepsin. All right, so excuse me. So pepsinogen is secreted by those chief cells. It's an inactive form of pepsin. And then we have hydrochloric acid, which are produced by the parietal cells, and that converts pepsinogen into pepsin. So pepsin itself, remember that it is formed from pepsinogen in the presence of hydrochloric acid. We say that it's an autocatalytic effect. And an autocatalytic effect or reaction is just a reaction where the product itself acts as a catalyst for the reaction. So in this case, pepsin would kind of stimulate the production of hydrochloric acid to increase the rate of reaction of converting pepsinogen to pepsin. And pepsin is just an active enzyme that's going to break down proteins into polypeptides. So, and we'll see, we'll go into more detail about how proteins get broken down later on, but remember, proteins are eventually going to get broken down into amino acids. So right now, pepsid is going to break down these big old proteins into polypeptides, which are essentially long chains of amino acids. And gastric lipase is going to be found in relatively small quantities. And they're fat-splitting enzymes. Their actions are going to be inhibited by low pH. So if we're talking about, the pH of gastric juice or gastric acid or gastric enzymes, remember that hydrochloric acid is a really strong acid, and our gastric juice typically is around a pH of 1.5 to 3. Somewhere around there. And if we add bicarb, remember we talked about bicarbonate ions that are going to increase our pH to about 5.5 to 6.5 increase our gastric acid to about 5.5 to 6.5. So when we convert pepsinogen to pepsin, in the presence of hydrochloric acid, we're really dropping the pH of our stomach acid to about 1.5 to 3, somewhere around there. And remember, when we break down molecules, so we have this active enzyme pepsin that's going to break down proteins into polypeptides, or a fat splitting enzyme. We're splitting up fat. That is a hydrolysis reaction. And remember, adding molecules together, the opposite of hydrolysis is dehydration synthesis. All right, so now what would happen, do you think, if you had a genetic mutation that prevents parietal cells from releasing hydrochloric acid? So a couple of things would happen. One, if you don't have this hydrochloric acid, one, you would not have pepsin. Two, you would not, pepsinogen would not be able to be converted into pepsin. Two, your pH of your gastric juice would remain elevated because we don't have that hydrochloric acid to lower the pH. And also, without pepsin, we would not be able to break down those proteins into polypeptides. So the same thing happens if you take a medication. Let's say you take Pepsid. I'm sure you've heard of Pepsid. Pepsid is a medication that's going to decrease your hydrochloric acid production in the parietal cells of the stomach to reduce symptoms of like a gastric ulcer, something like that. So remember, hydrochloric acid converts pepsinogen to pepsin. So this pepsid would lead to a lower level of hydrochloric acid. It would decrease the conversion of pepsinogen to pepsin. And then a decrease in that pepsin would eventually lead to a decrease in protein metabolism because we wouldn't be able to break down those proteins into polypeptides. We also have mucus cells. Remember, those mucus cells secrete mucus, and that's just going to provide lubrication for the stomach lining. We also have this thing called intrinsic factor that's also produced by parietal cells, and that's going to be important for vitamin B12 absorption. We're going to find this in the small intestine, the jejunum. The small intestine has three parts, the duodenum, ileum, jejunum. We'll talk about that when we get to the small intestine. Also, these chemical messengers, they're secreted by these enteroendocrine cells. They're hormones, local messengers, and that's going to help regulate the digestive processes overall. All right, so far, here's what we've talked about. We've talked about mucus, we've talked about pepsinogen, we've talked about pepsin, hydrochloric acid, chemical messengers, and intrinsic factor. And this is within the stomach. So this is gastric acid or gastric juice or digestive juice. And that's actually produced pretty much 24/7. The rate of production is going to vary. It's under nervous system control and it's under hormonal control. So remember, for the nervous system or neural regulation, we have the parasympathetic and sympathetic activity. And remember that sympathetic activity, when you meet the bear in the middle of the woods, your body doesn't care about digestion. So sympathetic impulses are going to decrease or inhibit gastric activity, whereas parasympathetic impulses are going to increase your gastric activity. It's going to promote the release of histamine, which is going to stimulate gastric secretion. And then, with gastric secretion, we're going to see, the conversion of pepsinogen to pepsin. We're going to see a lot of this metabolism of, or anabolism of, proteins and carbs and lipids and nucleic acids. All right, from a hormonal standpoint, remember, the regulation of gastric secretion is under neural and hormonal control. So under the neural is sympathetic, parasympathetic. Under the hormonal control, we have different enzymes. So we have somatostatin. Somatostatin is going to inhibit hydrochloric acid secretion. So somatostatin inhibits hydrochloric acid secretion. So if we inhibit hydrochloric acid secretion, think of pepsin. So we're gonna decrease our conversion of pepsinogen to pepsin, which over time could affect the digestion of proteins. We also have a hormone called gastrin, which increases the secretion by the gastric glands. And basically, it's going to stimulate cell division in the mucosa and of the stomach and small intestines. So, I already said that your pH of your stomach is about, 1.5 to 3.5. Typically, when we eat food, we don't eat a lot of, we don't eat battery acid. So we don't eat foods that are, 1.5 of pH. So when we eat food, it's going to raise the pH inside of our stomach. It's going to raise that, the stomach pH. And as the food mixes inside of our stomach, this gastrin is going to be released. And and then eventually, so this gastrin is going to get released as our pH as our pH rises. Because remember, our gastric acid likes to be at about 1.5 to 3.5. So we eat the food, and let's say our pH goes up to 6. So this gastrin is going to be released to help bring our pH down. So our body's going to release gastrin. Now our pH is 5, release more gastrin. Our pH is 4. Now release more gastrin. Our pH is 3. Oh, we're getting close to where our pH likes to be. So our body's gonna our body's gonna kind of slow down on our gastrin secretions. So our pH is now 2.5, 2. So our gastrin's really kind of shutting off a little bit. And then once our pH of our stomach acid is about 1.5, gastrin secretion is going to stop. So this is a negative feedback loop. If something is too high, we're going to lower it. All right, we also have this thing called cholecystokinin, or CCK. A lot of times we just say CCK. This is going to be released by cells in the small intestine whenever fats or proteins enter the small intestine. And what it does is going to decrease our gastric motility so that not too much food enters the small intestine all at once. So we have different mechanisms in place, and we'll talk about, stretch receptors and how stretch receptors allow the food to come in periodically into the small intestine. But another way is this CCK, or cholecystokinin, that's going to decrease the gastric motility. So if the small intestine says, hey, we have proteins and fats in here, the small intestine has work to do. So it's going to release cholecystokinin, and CCK is going to tell the stomach, hey, hey stomach, I'm the small intestine, I'm working right now. Don't give me any food. And it's just going to regulate those pancreatic secretions of digestive enzymes. All right, here's just a negative feedback picture on how gastric secretions are regulated. So remember that this is a parasympathetic nervous system. So we have those impulses are conducted through the parasympathetic preganglionic nerve fibers. Remember that this is the vagus nerve, cranial nerve number 10. Remember I said in chapter 10, 11, 10 or 11, one of those, that, the vagus nerve is one of the most important nerves of the body. The vagus nerve does a lot for our visceral organs. And then parasympathetic post-ganglionic fibers, those impulses are going to stimulate the release of gastric juice from the gastric glands, which are going to stimulate gastrin to be released into the bloodstream. And that gastrin is going to release more gastric juice. All right. Now, when we talk about gastric secretions, we have 3 different phases. And the celiac phase, the gastric phase, and the intestinal phase. So remember that, your stomach has a pH of about 1.5 to 3.5. And when you eat food, it's going to raise the pH inside of your stomach. As the food mixes in your stomach, gastrin will be released. Once the pH inside of your stomach hits around 3, gastrin secretion will slow down. And once the pH hits about 1.5, gastrin secretion will stop. All right, so in the celiac phase, this is where um, we might not have even taken in any food. We might not have even ingested food, but that sight of food, or that smell of food is going to start the whole process beginning. We might have gastric juice secreted in response to walking into grandma's kitchen and smelling whatever grandma's making at the time. And then in the gastric phase, this is when we have mechanical and a little bit of chemical digestion. We see that our reflexes are going to kick in, and we have gastric juice that's going to be secreted as well. And in the intestinal phase is when food actually goes from the stomach into the small intestine, and and um, and that gastrin is going to get shut off when, or that gastrin is going to be released in the presence of hydrochloric acid and, oh no, that's cholecystokinin. I'm sorry, CCK. CCK is going to get released when proteins and fats enter the small intestine. Gastrin is going to be released by the gastric glands to help keep your pH of your stomach low. All right, so for gastric absorption, remember there's a difference between absorption or digestion and absorption, or breaking down and absorption. So breaking down is not the same thing as absorption. So enzymes, so remember pepsin breaks down proteins. Lipases usually break down fats. We have in the stomach wall, we have that mucosal layer that's going to protect the stomach itself from, or protect the stomach from autophagy, or from self-eating, or from the gastric juice from eating away at the stomach lining. So the stomach itself does not do a lot of absorption. A lot of absorption happens in the small intestine. But the stomach does absorb some substances, such as water, some salts, some certain lipid-soluble drugs, and some alcohol. So the stomach's primary job is to mix food. Remember, we had the mixing movement and the propelling movement. Remember segmentation, that's going to occur, or churning. Segmentation occurs in the small intestine, but churning, that's going to be a mixing movement. So the stomach's primary function is to mix the food or churn the food, and to break down the food, some chemical digestion, so that we can break down those proteins into smaller particles. All right, so we have that bolus that we've swallowed, and that bolus plus the gastric acid forms this thing called chyme that we talked about. And chyme is just this semi-fluid paste thing. Remember that peristalsis happens from the pharynx all the way down to the anus. So peristalsis is going to push that chyme through the stomach, down towards the bottom of the stomach in that area called the pylorus. Remember, I had the pyloric antrum, which was like that waiting room. And then just a small amount of chyme is going to be transported through the pyloric sphincter at any given time. Remember, we don't want, the whole Big Mac to go from our stomach to the small intestine. We only want a little bit of Big Mac to go into our small intestine at any given time. And when that food gets to the pyloric area, these peristaltic waves are going to push the contents backwards and are going to help mix the food. And then remember, we had that lower esophageal sphincter, or that cardiac sphincter, up at the top of the stomach that connects the esophagus to the stomach through that hiatus, or the diaphragm, that esophageal hiatus, to prevent backflow of chyme or gastric acid into the esophagus. And the rate of emptying depends on the consistency of chyme and the type of food. So, if we didn't do a good job of mechanical digestion, if we just kind of swallowed our food whole, our stomach's going to need to do our stomach's going to need to work harder to break down that, or to do more mechanical digestion of that chyme before it can release that chyme into the small intestine. But and we'll talk about I said that there's 3 different parts to the small intestine. The first part's the duodenum, but as that chyme, or that food plus stomach acid, enters the small intestine, or the duodenum, this is where a lot of our secondary organs are going to kick in. We have the liver, the pancreas, and a lot of it's going to be the pancreas. But the pancreas is going to start to release chemicals to help further break down those food particles, so that eventually the small intestine can absorb those food particles. So as chyme starts to go from the stomach to the small intestine, or the duodenum, I mentioned earlier that we have these stretch receptors. Remember that cholecystokinin, or CCK, is going to detect that there's proteins or fats that are in the small intestine and say, hey, stomach, don't send me any more food because I'm still working down here on the food that you've just given me. In addition, we have these stretch receptors that are going to kind of help add on to that mechanism of not giving up food from the stomach to the small intestine. So these stretch receptors are going to initiate this thing called the enterogastric reflex that's going to slow food from going from the stomach to the small intestine, so that the small intestine can maximize its chemical digestion and its absorption. So here you can just see that, we've brought food down into the green is, I guess, the food. So we brought food down into the stomach, and the yellow is the gastric acid. We've mixed the food, and then in that picture on the right-hand side, that pyloric sphincter, I guess in the first 2 pictures, you can see the pyloric sphincter is closed, and then here we're mixing the food. And here we've opened up the pyloric sphincter and allowed some of that chyme to go into the duodenum, or the first part of the small intestine. And remember, all of this is stimulated by the vagus nerve, or the parasympathetic nervous system. From a hormonal control, we're talking about CCK, cholecystokinin. From a neural system control, a nervous system control, or a neural control, we're talking about the vagus nerve. So here, the pyloric sphincter opens, food goes into the duodenum. The duodenum fills with chyme. We have this duodenum is stretched so that we have those sensory stretch receptors are stimulated. That's going to go to the brain, and then up through the vagus nerve, to the CNS, and then it says a reduction in parasympathetic activity decreases peristalsis in the stomach wall, so that food can stay in the stomach until those stretch receptors stop sending signals up to the brain. All right, now heartburn. Even though we call it heartburn, it's not actually, your heart is not on fire. It's just the general area. Remember that we have referred pain. And typically we say that the pain, if we have heartburn, the typical pain is somewhere around your heart area. So, so that's why we call it heartburn. Typically, a lot of times we see that heartburn is a result of eating a lot of food too quickly. So, like I said, it does take about 20 minutes for the hypothalamus to sense that the stomach is full. And that fullness, if you eat too much food, you can lead to abdominal pain and gastric reflux. And that's when reflux is when that stomach acid or the stomach contents goes from the stomach and backflows into the esophagus. So that lower esophageal sphincter relaxes a little bit, or that pressure in the stomach is greater than the pressure exerted on that lower esophageal sphincter, and stomach contents actually goes into the esophagus. And the pH of stomach acid is 1.5 to 3.5 type of thing, and the esophagus really can't handle the acidity of that gastric juice, so it inflames the esophagus, and we call that esophagitis. And remember, it feels like heart pain, so we call it's a referred pain, so we actually call it heartburn. Some people can find relief through antacids. And if you look on the label of antacids, you'll see that antacids usually contain some type of sodium or aluminum, calcium, or magnesium, which is going to bind to those or neutralize those acidic portions. So it's not that the sodium neutralizes it, but usually the component that goes along with the sodium, aluminum, calcium, magnesium is what's going to bind with the hydrogen ion from the acid or the stomach acid. So ways that you can prevent heartburn, eat smaller meals, eat upright, eat slower, try and avoid caffeine as I'm drinking my coffee, avoid nicotine, avoid alcohol. One thing that I'll mention here is, I kind of briefly talked about it when we talked about Pepsid. And I kind of talked about Pepsid, but I didn't really mention ulcers. How we have that stomach lining, or that mucosal layer, and I mentioned that the mucosal layer is there so that the stomach acid doesn't wear away at the stomach lining. We have these things called peptic ulcers that are a result of kind of like a hole in that mucosal layer where stomach acid does kind of tear away at the stomach wall. And it can be a result of a bacteria called Helicobacter pylori or H. pylori. Um, so H. pylori can cause peptic ulcers. It's just a bacterial infection, usually treated with antibiotics and some sort of PPI or proton pump inhibitor that will decrease some of your gastric acid secretions. Another thing that I want to talk about is called a hiatal hernia. I'm going to go back up. Let's see if I want to do it right. No, I'll do it on this slide. So I'm going to erase something. I'll erase these. Okay, so a hiatal hernia, and I'll do it in green here. So we have this esophageal hiatus. So we have our diaphragm that's right here, and then we have our esophagus that comes down here. And then we have, our stomach, and we have this lower esophageal sphincter or our cardiac sphincter. And remember, if you know, a hernia is basically something that pushes through a muscular wall. We can have a hiatal hernia, we can have an incisional hernia, we can have an inguinal hernia. So we have several different types of hernias that we could have, but if we have a portion of the stomach that kind of bulges out through the throughout this or kind of bulges up through the diaphragm muscle, that's called a hiatal hernia. So a hiatal hernia is just a condition where the stomach pushes up through the diaphragm. That can actually cause severe heartburn. It's usually treated with surgery. It's a relatively, I don't want to say easy surgery because no surgery is easy, but it's a relatively lower risk surgery. Alrighty. Let me figure out where I was. All right, heartburn. Okay. All right, so vomiting. Vomiting is basically a reflex where we're going to empty the stomach contents in a reverse direction. It's called regurgitation. It can be caused by, certain drugs, toxins from food, an overstretching or over distension of the stomach, body motion changes, motion sickness associated with your inner ear. Basically, we have a vomiting center in the medulla oblongata, which is an area in your brain, in the posterior inferior side of your brain. And basically it's a motor response where, motors is going to be an action that's going to happen. It's a motor response basically where you're going to have that, deep breath. You're going to raise your soft palate, closing your nasal cavity, your glottis, relaxation of your lower esophageal sphincter, your cardiac sphincter. Your diaphragm contracts, your abdominal wall muscles contract, and you force fluid out through your mouth, or force food out through your mouth. And that's just the geeky way or the medical way of saying you puked, you hurled, you vomited all over the place. And not a fun thing to clean up. All right, so your pancreas. Remember that your pancreas has several different purposes. Your pancreas is part of your endocrine system in that it helps regulate your blood glucose level with, insulin and glucagon. And then your pancreas also has endocrine functions and exocrine functions. Remember that the hormones get released into your bloodstream directly, but also pancreas can deliver those hormones into the free apical surface or into ducts. And we'll see. We'll see on the next slide the structure of the pancreas, but we have these things called acinar cells or pancreatic acinar cells that are going to make up most of the pancreas, and these cells are going to release pancreatic juice into your pancreatic into ducts that are eventually going to go into the pancreatic duct. So pancreatic acinar cells are going to go into are going to release their contents eventually into the pancreatic duct. The pancreatic duct eventually will go into the small intestine. Now, the pancreatic duct is going to kind of hook up with this common bile duct, which is going to come from the liver and gallbladder. And it's going to, together, all three of these are going to empty acid into the small intestine, but more specifically, the duodenum area of the small intestine. And then we have the pancreatic duct and the common bile duct are going to join at this little tubule thing. There's this tube thing. It's a dilated tube called this hepatopancreatic ampulla, or this ampulla of Vater, like Darth Vader, Vater. Vader just means father in German. And we also have this hepatopancreatic sphincter, or the sphincter of Oddi, O-D-D-I. And it surrounds this ampulla. It controls the movement of bile and pancreatic juice into the duodenum. And the pancreatic itself is attached to the duodenum, and it secretes these fluids rich in digestive enzymes. So here you can see that the pancreas is going to lie kind of in the middle to left upper quadrant of your gut. Here you can see the we don't have the we're not really looking at the microscopic level, so we don't have these acinar cells, but basically we have these acinar cells that are going to release their contents into these, eventually going to make their way into the pancreatic duct. And from the liver, we're going to have this common hepatic duct with this bile duct, and eventually it's going to hook up with the pancreas, and they're going to release their contents through this thing. And now I guess we'll jump into this this blown up picture here. So we have the bile duct and the pancreatic duct. This is our hepatopancreatic sphincter. And remember, a sphincter is just a circular muscle that kind of controls the amount of gunk that gets released, or the amount of hormone that's going to get released into whatever space we're talking about. Or, we have the lower esophageal sphincter that's going to control how much food goes from the esophagus to the stomach. We have the pyloric sphincter that's going to control how much food gets released from the stomach to the small intestine. So here, this hepatopancreatic sphincter is going to dictate, the bile and the pancreatic juices going into the duodenum. And here we have this hepatopancreatic ampulla that is kind of like the convergence of the hepato or the bile duct and the main pancreatic duct. and it gets released into the duodenum of the small intestine. So the duodenum is that first area of the small intestine just after the stomach. So your pancreatic juice itself, so what gets released from the pancreas, um, has a bunch of enzymes that are going to digest carbohydrates, fats, proteins, and nucleic acids. So the pancreas does a lot. It has a lot of digestive functions. Um, and all enzymes that come from the pancreas are secreted in an inactive form. Remember, we talked about pepsin-ogen. So typically, anything that ends in ogen is the inactive form. Um, so we have pancreatic amylase is going to split up starch and glycogen into disaccharides. Remember, we had the salivary amylase that is going to split up that starch into disaccharides. All right, I guess I'll go. I forgot to mention, I guess I forgot to put it in the slide. So, an inactive form of something is called a zymogen. All right, so pancreatic amylase splits starch and glycogen into disaccharides and monosaccharides. Um, Pancreatic lipase um, is going to break down triglycerides or fats. Trypsin is released as trypsinogen. That's the inactive form. And trypsin is going to get converted by this thing called enterokinase in the small intestine. Um, so here you would have trypsinogen with the arrow going to trypsin, and the enzyme would be enterokinase. All right, so here you can see the trypsinogen going to trypsin with the enzyme or the catalyst enterokinase. The process is a little bit more involved. We're definitely not going to go into this, into that process in this class. But basically, trypsin is going to digest protein or break down proteins even further. We have a condition called acute pancreatitis, and that's seen with the conversion of trypsinogen to trypsin within the pancreas. And it's usually a problem with that conversion. It's usually caused by dysfunction or a problem with the enterokinase. So just know that acute pancreatitis is often caused by a defect with the conversion of trypsinogen to trypsin. All right, some more pancreatic juice enzymes. So chymotrypsin is released as an inactive form. It's activated by trypsin, and it also helps digest proteins. Carboxypeptidase is released as the inactive form. It's activated by trypsin as well. So you can see that trypsin does a lot of we remember that autocatalytic effect or autocatalytic reaction. So trypsin is the enzyme responsible for a lot of these subsequent reactions. So carboxypeptidase is also important in digesting proteins. All right, and I just added in here the inactive forms of chymotrypsin is chymotrypsinogen, Carboxypeptidase is pro-carboxypeptidase. All right, so we also have these things called nucleases, ribonuclease and deoxyribonuclease. They're going to digest or break down RNA and DNA respectively, which are digested in the small intestine. Remember that the stomach does a lot of mixing and doesn't do a lot of breaking down, does do some breaking down of proteins, but the duodenum is the big area for continuation of breaking down or chemical digestion. And then we also have bicarbonate ions. Remember, bicarbonate is HCO3. Bicarbonate ions, all they are there for is to help make that pancreatic juice more alkaline. It's going to raise the pH. Remember, 1.5 to 3.5, and then that bicarb is going to raise it to pH of 5.5 to 6.5 to help buffer that stomach acid. Some more pancreatic juice on the hormonal side that helps regulate the release of pancreatic juice. So these typically don't, these hormones don't break down stuff. These regulate the digestive enzymes that break down stuff. So secretin or secretin. Prosecretin or prosecretin is the inactive form, and secretin or secretin is going to stimulate the pancreas to secrete or release pancreatic juice that are high in the bicarb ions, which is going to make it more alkaline. I'm going to put more alkaline as opposed to alkaline, because alkaline, remember, is pH above 7. So typically we're going to go up to about 6.5. So we'll just say more alkaline as opposed to alkaline. Cholecystokinin, remember, that is going to be released when proteins or fats enter the small intestine to tell the stomach to, hey, stop sending food from the stomach to the small intestine. So it's going to regulate the pancreatic secretion of digestive enzymes, the pancreatic juice, nervous system regulation. Remember, we had the cephalic and the gastric phases. That's under parasympathetic control. It's going to stimulate the pancreas to secrete digestive enzymes. So with prosecretin or prosecretin, it's the inactive form of secretin. If you have a problem with prosecretin or prosecretin, whatever, so what happens if you, so we'll talk through this. So secretin, what secretin does is it's going to stimulate the pancreas to release bicarbonate. So secretin is going to increase your pH. If you have a problem with prosecretin, therefore you're not going to convert prosecretin to secretin. So you're not going to get that increase in pH. So if you have a defect with prosecretin, you're going to have a decreased pH in your stomach acid. So therefore, if you have a problem with prosecretin, alkaline pancreatic juice will not be released from the pancreas when chyme enters into the duodenum. And you can see peptic ulcers develop as a result of that. All right, here is just the hormonal regulation of Pancreatic secretion, I'll just write hormonal there. You can see the acid or the acidic chyme enters into the duodenum. And then that pancreatic juice is going to have a lot of bicarb to counteract that acidic. Oh, let me go in order. So we have that acidic chyme that enters the duodenum. And then we're going to have secretin is going to get released into the bloodstream, which is going to release bicarb to increase that pH level to help neutralize that acidic chyme. All right, the liver. Liver is one of the accessory organs. It's the largest internal organ in the body. It's located in the upper right abdominal quadrant, just below the diaphragm or just inferior to the diaphragm. It's a reddish brown organ. It's very, very vascular, has a lot of blood vessels. It's not part of the GI tract. And we can see that it has four lobes. There's the right lobe, which is the biggest lobe, and then it has the left lobe, which is a little bit smaller than the right lobe. We have the quadrate lobe, which is it's also called a minor lobe, which is going to be near the gallbladder. And then we have this little lobe down here called the caudate lobe, and it's going to be near the inferior vena cava. Remember, the inferior vena cava is going to be what returns blood to the right side of the heart, the right atrium. And the lobe, or the lobes of the liver, if we look at it more microscopically, or kind of look at it, break it down, we'll see that these lobes have, these lobule things, and the lobules are the functional units of the liver, and they're going to contain these liver cells called hepatocytes around a central vein. And then all right, so I like the picture version better, so I'm gonna advance the slide to the picture, and then I'm just gonna let you know. So we have these lobule things. The liver has these lobules, and so here, down here are the lobules. All right, so like here you can see the lobules. Here you can see the lobules, and they contain these hepatocytes. And the hepatocytes basically are just liver cells. So I'm just looking real quick. Okay, so like that's a liver cell, that's a liver cell, that's a liver cell, that's a liver cell. So we have these hepatocytes that are that's going to lie around this thing called a central vein. There's your central vein, and the central vein is going to be we'll talk more about it later on, but it's used for like your central drainage system. We also have these hepatic sinusoids that are between these plates of cells. Here's where your sinusoids are, and it's going to help drain a lot of your bad gunk to the central vein. So it's like it's also part of the plumbing system. We have the hepatic portal vein, which brings nutrients to the sinusoids. So here we have the hepatic portal vein, which is going to bring the blood to the liver for processing. Now, remember that the definition of a vein is typically anything that brings blood to the heart. The liver, and we talked very briefly about how the liver has this exception to the rule, and I said we talk about that when we got to the digestive chapter. We'll talk about this first hepatic metabolism, our first pass hepatic metabolism. So not all veins carry blood to the heart. So this hepatic portal vein will carry blood to the liver, typically from the stomach. So this hepatic portal vein will bring nutrients from typically the stomach or small intestine to the liver, specifically to the sinusoids, or these spaces between the cells. And then as well, this hepatic artery, the hepatic arteries themselves, arteries typically carry blood away from the heart. But arteries also carry oxygen-rich blood, with the exception of the pulmonary circuit. But hepatic artery is going to bring oxygen-rich blood, which is eventually going to mix with this oxygen-poor blood from the portal vein, and this blood is going to flow in and around all of these sinusoids. So here you can see, the blood's mixing with in the sinusoids, and it's going to kind of run in between all of these hepatocytes, and then eventually they're going to go to the hepatic into the central vein. And then to the hepatic duct. So you can see that down here, you have your hepatic artery is going to go, and your hepatic vein is going to go to the central vein, which is the big blue one. And that central vein is eventually going to go to the hepatic vein, and then we're going to go to the heart. All right, so what does the liver do? The liver does a lot. So the liver, remember that the liver can store glucose in the form of glycogen. So you eat your pasta, you use the glucose that you can use, and then the liver, if we need to store glucose, the liver will convert that glucose into glycogen and store it. So because we are creating glycogen, we call that glycogenesis. Genesis means to create, glycogen, so glycogenesis is creating glycogen. And then tomorrow we need that energy that we need that energy because we're running the track meet, or we're doing the swim meet, or that football game for that pasta that we ate the night before. So now we need to take that stored glucose, or that stored glycogen, and we need to convert that glycogen back to glucose to use for energy. So remember, lysis means to break down. So we're going to take that glycogen and break it down. So glycogenolysis. Glycogenolysis is to break down glycogen and convert glycogen into glucose. Remember that, typically carbs are going to get digested and broken down into glucose, but we, if you look at, diets that don't don't have carbohydrates, or you limit your carbohydrates, your body can find other ways to get glucose. And we can do that by using non-carbohydrate sources to get glucose. And that is gluconeogenesis. So gluco is glucose, neo is new, and genesis means to create. So we are creating new forms of glucose, gluconeogenesis. Oxidizing fatty acids. So we're going to take fatty acids and so synthesizing lipoproteins. So this is all your cholesterol panel or your lipid panel that you get when you get your blood drawn. Your lipoproteins are your HDL, LDL, VLDL, stuff like that. Phospholipids, cholesterol. And then also your liver will take that excess carbs and proteins and convert those into fat. So anytime that your body or anytime that you take in too much of something, your body will convert that into fat. If you eat too much pasta and, your body's going to use that energy for right now, and then your body's going to convert whatever it can into glycogen and store it in the liver and the skeletal muscle cells. But your body can only hold so much glycogen, and the rest of the carbs that you ate will get turned into fat. Deaminates amino acids. We'll talk a little bit. If you don't remember the structure of amino acids, we'll talk about the structure of amino acids later on in this chapter. But there's an amine group of on amino acids, and the liver is going to remove that amine group. Forms urea. Urea is going to be one of the components of urine. We'll talk about that when we get to the kidney chapter. Synthesizes plasma proteins. So remember, plasma proteins are going to be what's found in your bloodstream. A lot of your albumins, your gamma globulins, that's going to be in your bloodstream. That's going to come from your liver. Converts amino acids into other amino acids. So remember, we have 20 amino acids. And if your body's good about, if you're deficient in one area, we can convert, this amino acid into this amino acid because we need more of this amino acid, and we have too much of this amino acid. Stores glycogen. We've already talked about that. Stores iron, vitamins A, D, B12. Remember, your fat soluble vitamins are A, D, E, and K. Stores iron. We talked about storage of iron in the lymphatic system chapter when we talked about getting a fever, and when we get a fever, we remove that iron component out of heme to prevent viral replication and bacterial replication. We store that iron in the liver. The liver does a lot with phagocytizing damaged red blood cells and foreign substances. Removes toxins such as alcohol and certain drugs from the blood. Remember that I said that the stomach will absorb, some water and some alcohol, and then I said, I've talked a little bit about first pass hepatic metabolism. We'll talk about that here in a few minutes. So this bloodstream from the stomach will go to the liver. And the liver will process the alcohol that was absorbed through the stomach. But cirrhosis of the liver can result from, long-term alcohol use or long-term drug use. Acts as a blood reservoir. So the liver, remember I said, is highly vascular, has a lot of blood vessels. So injury to the liver, if you get in a car wreck and if you nick your liver, you can have a serious um, bleeding issue. Produces and secretes bile. A lot of people think that would fall under the gallbladder, but it doesn't. Um, so, um, know the functions of the liver. And so if you also know what happens if something is going wrong with the liver. So if you, for example, if you have an injury to the liver, you might have uh, a, an issue with lipid digestion. Because remember, your liver does, well, first it synthesizes your lipoproteins, but your liver converts non-carbohydrate sources into glucose. So if you have damage to your liver, maybe we have a defect here, or oxidizing fatty acids. So injury to your liver can result in lipid digestion being affected. All right, here's just a summary slide of not necessarily a summary slide, but more of an expanded slide of what we talked about. Detoxification is removing toxins from the liver, carbohydrate metabolism, lipid metabolism, protein metabolism, storage, blood filtering, secretion. Obviously, the liver, based on this previous slide, or the previous two slides, the liver is very, very much required to maintain life. And what we found is that the liver is actually capable of regeneration if at least 25 to 30% of it is healthy. On the downside, or on the negative side, if you do get liver cancer, it's very serious. Liver cancer doesn't have a good prognosis, and typically, you only have weeks to months with liver cancer. But we do have donations. People can donate a part of the liver, because as long as if you are a donor, and as long as you keep 25 to 30% of your liver, if you donate half of your liver, your liver can regenerate. But people don't want to donate their organs, so there are shortage of donors out there. We also have these things called ELADs, or extracorporeal liver assisted devices. Extracorporeal just means outside the body. And that just, we have like dialysis for kidneys. This is just kind of like dialysis for the livers. It's an artificial liver that has human liver cells in it. And it's kind of like dialysis. Let's see. But I will say the process and the machinery is much more complicated than a dialysis machine. But basically, you're going to take the blood from the patient, run it through this ELAD machine. Toxins are going to be removed, and then the plasma is returned to the patient. All right, so bile. Remember that I said bile is synthesized and stored in the liver. Bile is just this yellowish green substance liquid that the liver cells, or hepatocytes, secrete. We'll start using some medical terminology. So components of bile, water, bile salts, Hepatic cells are going to use cholesterol. Cholesterol is typically the main component to bile salts. So if you eat a lot of fatty foods, your bile salts, or you have an increased risk of forming gallstones. Um, so bile salts are going to be used for to emulsify fats, and we haven't really talked about emulsification yet. Um, and it says here only bile salts are the only bile component that have a digestive function, and they act as fat emulsifying agents. So remember that our bloodstream is water soluble or hydrophilic, and fat and water, they don't mix. So our fat is hydrophobic or lipophilic. And in order to transport fats throughout the bloodstream, we can't just have fats free floating in the bloodstream because of the hydrophilic and hydrophobic and the water soluble and the lipid soluble. So fats have to be kind of engulfed in order, or kind of have to be surrounded or emulsified in order to be transported around the bloodstream. And we'll see this in a few slides. So bile also has these bile pigments called biliverdin and bilirubin. And it's derived from red blood cell hemoglobin breakdown. Remember that when we break down red blood cells, we break them down into the biliverdin, and then it's further broken down into bilirubin. Cholesterol is one of the big components of bile. Electrolytes, sodium, potassium, calcium. We do see jaundice sometimes. So jaundice, you can see the eyeball here. It looks very yellow. It's just an abnormal yellowish tint of tissues, and it could be your skin or your eye, the sclera of the eye, or your mucous membranes. It's just an abnormal accumulation of bile pigments. And it can be caused by several different things. It could be an obstructive cause. Let's say that you have a bile duct occlusion, and you have you have an obstruction of your bile duct, and your bile is kind of leaking out, and so that could cause jaundice. It could be at the cellular level because you have cirrhosis or hepatitis. It could be because of a hemolytic process, because of your red blood cells are being rapidly destroyed. Not so much today because of transfusions, because we have a lot of additional protective mechanisms. But let's say if you have a blood infection that is destroying your red blood cells, or like hemolytic anemia. All right, so hepatitis. Hepatitis just means inflammation of the liver. Typically, we associate hepatitis, with hepatitis A, B, or C. We actually have A, B, C, D, E, F, and G. In the United States, we only see really A, B, and C. More Europe and definitely in third world countries will we see the D, E, F, and G. But we can classify it as acute or chronic. Acute is when hepatitis comes on in a severe way. I didn't have it yesterday and I had it today. Chronic is the symptoms slowly progress. Hepatitis may typically, when we think of hepatitis, we think of the hepatitis A, B, C, but it doesn't have to be that way. And I use myself as an example. I used to work in the emergency room before I went to nursing school, and I was working a shift. I was working night shift at the time, and during my 7 p.m. to 7 a.m. shift, I just started getting this, uh, basically pain in my liver area, and working in the ER, you have to have the hepatitis A and B shots, the vaccines at the time, and I had both of those, so I knew I didn't have Hep A and Hep B. But after I got off, I checked myself into the emergency room, and they tested me for A, B, and C, and basically I was diagnosed with hepatitis, or inflammation of the liver. But I didn't have infectious hepatitis like A, B, C, D, E, F, G. I just had, they called it idiopathic hepatitis. They couldn't figure out why I had hepatitis, but I had inflammation of the liver. And my liver, if you drew when you drew my blood, my liver panel was, throughout the roof. My ALT, AST, and my ALK phos were really, really elevated. But some people have symptoms. If you have hepatitis A, B, C, D, E, F, and G, you might have symptoms and you might not, but you are still carriers for the disease. And some of the infectious forms of hepatitis are bloodborne, meaning that you are that you have to have blood contact in order for them to be transmitted, and some other forms are transmitted by sexual contact or contact with other bodily fluids, or feces. If you have a viral form of hepatitis, remember that antibiotics are not effective against viruses. So for hepatitis A, that's going to be more of your food-based hepatitis, or contaminated with fecal matter containing hepatitis A. So that's usually seen sometimes in fast food restaurants, where employees, go take a dump and don't wash their hands, and they come back and transmit hepatitis A into the food. Or hepatitis B is seen with sexual activity or sexual contact, where the person is infected because they came in contact with body fluids that contain the hepatitis B virus. Hepatitis C is blood contact, and hepatitis C is the most common form of hepatitis. Hepatitis D, as in dog, you basically have to have hepatitis B, as in boy, to have hepatitis D, as in dog. And again, we don't see hepatitis D, as in dog, in the United States, really. Hepatitis E is very similar to hepatitis A, like the water in the food. But that's going to be found in more like your third world countries. Hepatitis F is water in food as well. Hepatitis G is usually sexually transmitted. It's usually found in combination with hepatitis C. And then I said, there's non-infectious forms of hepatitis, such as, from alcoholism, medications, immune reactions. You could have, this thing called alpha-1 antitrypsin deficiency. So, in my case, they asked me, are you an alcoholic? I'm like, no. And they did, ask me what meds I was on, and basically there was nothing that they could pinpoint as to why I had hepatitis. So that's why they just call it idiopathic hepatitis. All right, so your gallbladder is a pear-shaped sac, kind of looks like a pear. It's found on the inferior side or on the underside of your liver. You can see that coming off of the gallbladder, we have this thing called a cystic duct. And that cystic duct has valves in it to prevent backflow. And eventually the cystic duct will merge with the hepatic duct to form the common bile duct. And sometimes it's called the common hepatic duct. Common hepatic hepatic duct or the hepatic duct to form the common bile duct. And then that's eventually going to go to the small intestine. All right, so the gallbladder, remember that the liver produces and stores bile. But the gallbladder is where the bile salts are going to be stored and concentrated. Bile pigments and cholesterol is going to be stored and concentrated in the gallbladder. And then that bile is going to get released into the duodenum of the small intestine. Remember, through that common bile duct, if you remember that hepatopancreatic ampulla. And then we have that hepatopancreatic sphincter that's going to release the bile into the duodenum. And one of the complications that we see is gallstones. We'll talk about gallbladder disease in a couple slides, but gallstones is a result of basically sometimes, usually more often than not, it's cholesterol pigments that forms these solid crystals, and they can enlarge and form these gallstones. And basically it's excess bile concentration or too much cholesterol that has been secreted by the liver or an inflammation of the gallbladder. And remember that CCK is released when proteins or fats enter the small intestine to tell the stomach to stop sending food from the stomach to the small intestine. So CCK, remember, so the gallbladder does a lot, cholesterol, fat. So CCK also works on the gallbladder to simulate contraction of the gallbladder because fats, CCK works with fats, and cholesterol is a lipid soluble substance, so it falls under the fat or the lipophilic substance or the water insoluble substance. So CCK will control bile release. And here we can just see I'll do the hormonal regulation of bile release by CCK. Hormonal regulation of bile release by CCK. There we go. I'll just leave it like that. All right. So here you can see, we have that chyme that is going to enter the duodenum, and that, since we're talking about CCK, that's proteins and fat. But since we're talking about bile, we're going to be talking about fat. And then the cells are going to release CCK into the bloodstream. And then that's going to tell the stomach to, one, not release any more substances, but also that CCK is going to go up to the gallbladder and say, hey, I need you to secrete some bile. And then bile is going to go down into the cystic duct, into the bile duct, and into the duodenum through the hepatopancreatic sphincter. And into the duodenum as bile. All right, so here is, remember I said that term emulsify or emulsification, and that fats can't be transported in the bloodstream by themselves because of the hydrophobic and the hydrophilic and the lipid soluble and the water soluble. So bile salts are going to aid digestive enzymes through the emulsification of fat globules. So what's going to happen is, you eat your Big Mac, which is going to have a lot of fat in it, and that fat's going to get broken down into these smaller droplets or smaller fat globules. and then they're going to get surrounded um, by these things called micelles or micelles. Um, uh, where's my cursor? By forming these micelles. And so remember, lipids are fats. And this, remember that our cell membrane has, the hydrophilic side and the hydrophobic side. So that, you can kind of think of this as, our cell membrane is a lipid bilayer. This is just a one-sided, so it's a unicellular layer. So we have our hydrophilic side on this side, and then our hydrophilic on this side and our hydrophobic on this side. Or we could say lipophilic and lipophobic. And basically the fat side is going to line up with the fat side, and the water side is going to line up with the water side. So this would be our plasma. Actually, I'll just do our extracellular fluid. So that's our plasma. And this is how fats are transported throughout our bloodstream. So it's going to enhance, these bile salts are going to enhance the absorption of fatty acids and cholesterol by forming these things called micelles or micelles. And remember, your fat soluble vitamins are A, D, E, and K. And then once your bile salts are done transporting fats, they are going to be recycled and used again. All right, so we talked about gallstones before. Gallbladder is a disease. So some of the symptoms, pain in your right upper abdominal quadrant. It could go to your shoulder. Remember, that's referred pain. You could have nausea, vomiting, sweating. Let's do nausea, vomiting, sweating. You can do an ultrasound of the gallbladder, or an x-ray called a cholecystogram to look for gallstones. And gallstones can be found either in the gallbladder itself, in the cystic duct, in the hepatic duct, or the common bile duct. And if you do find gallstones that are present, sometimes you don't do anything. If the patient is experiencing hardcore symptoms, you might elect to do a cholecystectomy, which is a surgical removal of the gallbladder. And typically nowadays we'll do a procedure called a laparoscopic cholecystectomy, where they're going to do like 3 holes into your stomach area. And they'll put some cameras in there and kind of blow up your stomach with carbon dioxide gas, and so you're going to have like some incisions about that big, and they're going to cut out your gallbladder and take out your gallbladder. And another thing that they can do is called an endoscopy, to remove stones from certain areas. So if you've had a colonoscopy or an EGD, that's what we're talking about. I mean, not a colonoscopy, but we can do an endoscopy. Remember, obviously this is under anesthesia, so we'll put a tube down into your mouth and into your stomach, and we'll actually kind of go backwards. If you remember that hepatopancreatic ampulla and that hepatopancreatic sphincter, remember that sphincter of Oddi that I mentioned earlier? We could actually put that camera and do the endoscopy and actually go back up into your bile duct that way. and look at either like your cystic duct, or go up into your common hepatic duct, and then up into your right hepatic duct, and remove gallstones that way. And if we can't remove your gallstones, then we might have to elect to do a cholecystectomy. All right, so, and here are just some pictures of some different gallstones. I was actually out for a birthday dinner on a Saturday night, and I decided to eat some oysters. And on Sunday morning, about 6 a.m., I woke up, and so this was Saturday night into Sunday morning, and I woke up and I was doubled over in pain and started puking, and I ended up going to the emergency room, and found out that I had a bunch of gallstones, and I had to have my gallbladder removed. And because I was having active symptoms, they weren't going to do the gallbladder that day. They had to do the lap chole that day. They had to wait until my gallbladder kind of settled down before they could remove it. And they scheduled my surgery for the Tuesday, so in 48 hours, and they gave me the option of going home and coming back, or just staying in the hospital. I'm like, well, I'm going to stay in the hospital. So I ended up having my laparoscopic cholecystectomy on that Tuesday. And They said that I had 72 gallstones that they counted when they removed my gallbladder. And afterwards they asked, if I had, if I worked with, a bunch of radiation, or if I was and they really quizzed me on my alcohol use. And I'm like, I was, 25 at the time, I think. And I'm like, well, what the only radiation I dealt with was like in physics lab in college. And so they really couldn't figure out why I had a bunch of gallstones, but I had the gallbladder attack because of the iodine reaction from the raw oysters that I ate. And just as a side note, while I was in anesthesia, or having my laparoscopic cholecystectomy, they ended up nicking my bile duct. And on the day that I was supposed to go home, so I had my surgery on Tuesday, and then on Thursday I woke up and I was jaundiced all over. I was very yellow. They did a procedure where they looked at, it's called a HIDA scan, and they found out that I was leaking bile inside of my body. And then they tried to do this endoscopy thing. It's called an ERCP, endoscopic retrograde cholangio pancreatography, where they go in through my mouth and my stomach and up through that sphincter of Oddi to see if they could repair the damage. But because of the clips that they had used for the gallbladder lap chole, they couldn't get to the leak because of the clips. So they ended up having to open me up. And so now I have a scar about that big on my belly. Anyway. All right, so here is just a kind of a summary. It seems, a long time ago that we talked about, the gastrin and the somatostatin and the CCK and secretin. So just be familiar with these. All right, so the small intestine, comes off of the stomach through at the pyloric sphincter, and it connects the stomach to the large intestine. And it's going to be the majority of your abdominal cavity. And it's going to receive that chyme from the stomach, but it's also going to receive, remember, those pancreatic secretions, as well as those secretions from the liver. And The small intestine also is going to remember all of those pancreatic enzymes get dumped into the duodenum. So we're going to complete the, I should say, complete chemical digestion of the nutrients in chyme. And then we're going to start the absorption of everything that we have now digested. Remember, the stomach only does very little absorption, some water, some alcohol, some ions type of thing. But the majority of absorption is going to happen in the small intestine. And then, whatever's not absorbed in the small intestine is going to go to the large intestine to start the whole feces formation. And we can divide the small intestine into three parts: the duodenum, jejunum, and the ileum. The duodenum is the first portion. It's the smallest portion. It's the shortest portion of the small intestine. This is where most of your chemical digestion is going to take place. The jejunum is the middle portion of the small intestine. It's going to be thicker than your ileum and more active than your ileum. A lot of your absorption of your nutrients is going to take place in your jejunum. This is, absorption is jejunum. So think of absorption, think of jejunum. And the ileum is that last portion of the small intestine, or the distal portion of the small intestine. And that's where we have these things called Peyer's patches. Peyer's patches are just kind of like clusters of lymph nodes or lymphatic nodules. And also bacteria reside there. And in the ileum, this is where a lot of absorption of your vitamins and minerals are going to be. And this bacteria that lives in your gut or that lives in your small intestine or the ileum area, that's called your microbiome. So definitely be familiar with that word, microbiome. And this bacteria, it's not a bad thing. It's not necessarily a bad thing. It's a normal process. So it's normal for bacteria to be in this area. That's your microbiome. All right, so your jejunum, your second area, and your ileum, the third area, has this double layer fold called a peritoneum, also called your mesentery. So your mesentery, your peritoneum, and that's going to be interwoven with a bunch of blood vessels and lymphatic vessels and nerves to a lot of your intestinal walls. So because we have that peritoneum, that double fold, we call it a peritoneal membrane, which also has another name called the greater omentum. And that greater omentum or peritoneal membrane is just like a covering that's over the stomach, over those small intestines, the ileum, jejunum, and over the large intestine. So here you can just see kind of like the picture of the greater omentum. It's just kind of like this broad sheet of connective tissue that's just like the layer of sheet. All right, so intestinal wall, or the small intestine. So we still have these tiny projections called villi. Singular is a villus, plural is villi. Remember we had those four layers, inner to outer. And we talked about how they increase the surface area so that the food particles can kind of really dig themselves into to increase the absorptive area. or to increase the surface area for absorption of the um, chemical digestive end products. Um, and this layer is actually a simple columnar layer, simple columnar epithelium layer. And we want it to be a simple layer because we only want things to diffuse through one layer of cells. We don't want things to have to diffuse through multiple layers or a stratified layer. And then inside the villi, we have the blood vessels and the lymphatic system as well, or lymphatic capillaries. We call these lacteals. This lymphatic capillary is called a lacteal. And of course, there are nerve fibers that run through there. And all these cells are replaced, every 3 days to 6 days or so. Remember, as food passes through those, or as now we call it chyme, and as chyme passes through those, it actually tears away at that epithelial layer. So we do need to have kind of constant replenishment of those cells. And we also have these they're not on here. Let's see if they are on. No, they're not on here. But we also have these things called intestinal glands, or the crypts of Lieberkühn. They're located between here, these intestinal glands. They're kind of like another abyss or another pocket. They're called crypts of Lieberkühn, or intestinal glands. Let's see, plicae circularis, they're just circular folds of the mucosa that also increase the surface area. We'll see that on the next slide. So here you can see the plicae circularis. They're intestinal folds that allow for increased surface area. You can see the you can see not only the plicae circularis and the villi. So we have like multiple projections upon multiple projections just to increase that absorptive area. We have the muscularis layer. Remember, we still have those 4 layers: the mucosa, serosa, submucosa, muscularis. All right, intestinal epithelium. Not much to say here. Microvilli is there for absorptive purposes. All right. All right, so secretions of the small intestine. So we know we have mucus that are going to be secreted by goblet cells. And they're also secreted by these things called Brunner's glands. They secrete this thick alkaline mucus in response to certain stimuli. And these Brunner's glands, basically they protect the mucosal layer from the acidic contents from entering the duodenum. Also secrete this watery fluid that's from the intestinal glands to pick up the digestive products and transport them to the villi. They do not contain any digestive enzymes. We also have brush border enzymes, or just enzymes that we also call them brush border enzymes, that are in the membranes of the microvilli. We have peptidases that are going to break down peptides into amino acids. Um, Sucrase, maltase, lactase. Lactase are going to break down those disaccharides into monosaccharides. So sucrase will break down sucrose, maltase will break down maltose, and lactase will break down lactose. And we'll see that here in a few slides. Lipase is going to break down fatty acids into glycerol and the fatty acid side chains. All right. We also have these chemical messengers. Remember secretin or secretin in that CCK, secreted by the enteroendocrine cells. We also have an antimicrobial substance that are secreted by intestinal glands that are going to help defend against bacteria. Here's just a little chart that that kind of tells what is secreted where and what happens. Remember that, trypsin converts chymotrypsin into chymotrypsin, or trypsin converts chymotrypsinogen into chymotrypsin. Here, this, where is it? Enterokinase. Enterokinase, and sometimes you'll see enterokinase or enteropeptidase. They're going to be the same thing. Lactase. Lactase breaks down lactose. We talked about, you've heard of lactose intolerance. Just a side note, being lactose intolerant is actually genetically normal. So those of us who are lactose tolerant, so those of us who can tolerate lactose are not genetically normal. So people who are lactose intolerant, lactose cannot be broken down into those monosaccharides. Lactose actually breaks down into glucose and galactose. So this lactose sugar just kind of sits there, and the bacteria come in and basically feed on the sugar. And because remember, bacteria love sugar. And once bacteria feeds on the sugar, the bacteria is going to release these gases, and these organic acids are going to and these gases build up, and that's why you get the bloating and the intestinal cramps and the diarrhea if you are lactose intolerant. All right, so regulation of small intestine secretion occurs by one of these methods, or by these methods. So mucus secretion occurs in response to mechanical stimulation or presence of irritants such as gastric juice. Or the contact with chyme stimulates goblet cells to secrete mucus and intestinal glands to secrete watery fluid. Distension of the intestinal wall activates those nerve plexuses in the wall of the small intestine. Remember that vagus nerve. And the distension also stimulates the parasympathetic reflexes that trigger the release of intestinal enzymes. So nothing really new here. We've kind of already talked about those. All right, absorption in the small intestine. Remember that villi is there to increase the surface area for absorption. The small intestine is where absorption happens. Remember, not in the duodenum, because that's where chemical digestion is still taking place. But remember, the ileum is the big component for the jejunum is the big place where most of your absorption of nutrients takes place. And then absorption basically is so effective that by the time you get to the ileum, not much is left. not much food material is left. And there's a difference between, well, I'll just say, malabsorption is malabsorption is you can still, I'll just put chemically, digest or break down the nutrients. However, the blood supply cannot absorb what you've broken down, and you can experience abdominal pain. So malabsorption is when you can break down things, but you can't absorb them. So in my lactose intolerant example, that is not malabsorption. That's an issue dealing with chemical digestion. So malabsorption has nothing to do with chemical digestion. It's an issue dealing with absorption. Oh, I'm sorry, I didn't advance your slide, crud. All right, there you go. All right, so carbohydrates. I've said many, many times now, carbohydrates are going to get broken down into glucose. So we have salivary amylase and we have pancreatic amylase. We've already talked about those two. They are going to break down starch and glycogen into disaccharides, and I'll put and mono. Well, we'll just leave it as disaccharides. All right. So, and then once we get to the disaccharide level, we have these things called intestinal enzymes, and those are going to break down the disaccharides into monosaccharides. Those are going to be your maltase, sucrase, and lactase. And so monosaccharides, when we get to the monosaccharide level, those are going to be absorbed by facilitated fusion and active transport into the blood vessels and through the villi. Not that you need to, well, you kind of need to know. You need to generically know this chart. So, we have this huge carbohydrate here. And so basically we have this huge carbohydrate, and we have salivary and pancreatic amylase that's going to break down starch and glycogen into disaccharides. And we have three different disaccharides. We have sucrose, lactose, and maltose. And sucrose is basically glucose and fructose. Lactose is galactose and glucose, and maltose is glucose and glucose. So then sucrose, we have an enzyme called sucrase. Sucrase is going to split up glucose into, or sucrase splits sucrose into glucose and fructose. Lactase splits up lactose into galactose and glucose. And maltase splits up maltose into glucose and glucose. So really we are left with, when we break down disaccharides into monosaccharides, we are left with these three monosaccharides: glucose, fructose, and galactose. Now, we know that the body likes to use glucose for cellular metabolism, glycolysis, and we know that the nervous system, the central nervous system, can only use glucose as its energy source. We know the rest of the body can do the gluconeogenesis and convert non-carbohydrate sources into into energy. We learned that as one of the functions of the liver. So what will happen is the body will transform fructose into glucose, and the body will transform galactose into glucose. And now we have glucose as our end product. So eventually, basically we have carbs are going to go to glucose by way of salivary amylase, pancreatic amylase, and intestinal enzymes, sucrase, lactase, and maltase. And that is carbohydrate digestion. Let's do carbohydrate chemical digestion. And maybe we should get rid of absorption because we haven't really absorbed anything yet. All right. All right, protein digestion and absorption. So we know that pepsin is the ultimate end goal, and that pepsin is going to break down proteins into polypeptides. And, you know, we have the pepsinogen that in the presence of hydrochloric acid will convert pepsinogen to pepsin. And that pepsin will break down proteins into polypeptides. And then we also have the pancreatic proteases, you know, the trypsin, the chymotrypsin, that will break down proteins and polypeptides into smaller peptides. And then we also have the intestinal peptidases called carboxypeptidase that will break peptides into amino acids. And then amino acids are going to be absorbed by active transport into the blood vessels of the villi into the bloodstream. So here, remember that we had the, I talked about the amine group and deamination. So basically the amine group, that's this group right here. That's your amine group. That's your amine group that I was talking about. All right, so here we have a dipeptide, so two peptides. And remember, we have that dipeptide bond that's right here. And then remember, if we are breaking down something, that is an anabolic reaction. No, I'm sorry. Catabolic reaction. I think I might have said that wrong early on in the beginning. Breaking down something is a catabolic reaction. Catabolism is breaking down something. So we are going to add water into this reaction to split up that. We're going to break that peptide bond, and we're going to create two amino acids out of this. So here, and here I'll get rid of protein digestion and absorption because really all here we're doing is protein digestion. So we have this huge protein structure. And first we have, you know, and I didn't put in all of the, you know, the pepsinogen with the hydrochloric acid. So I didn't put that in here. So we have pepsin in the stomach that's going to break down proteins into polypeptides. And remember, polypeptides are just very, very, very long chains of amino acids. And then we have this enteropeptidase that's going to convert trypsinogen into trypsin. Trypsin is going to convert chymotrypsinogen to chymotrypsin. And then trypsin and chymotrypsin basically is going to take these larger polypeptides and break them down into smaller, more manageable chains of amino acids. So we're going to take this big picture, this big polypeptide chain, and we're just going to break it up into more manageable parts. And so 1, 2, 3, 4, 5, 6, 7, 8, 9, whatever. So we're going to have these more manageable parts, these smaller peptides. And then we have this carboxypeptidase that comes in, and we're going to break these carboxypeptidase. This carboxypeptidase is going to come in and cleave these peptide bonds to create the individual amino acids. So here we could say the carboxypeptidase in conjunction with water. So here we would need 1, 2, 3, 4, 5. We would need 6 water molecules to break up. Actually, we would need 5. We would need 5 water molecules. 1, 2, 3, 4, 5 water molecules to break up this smaller peptide chain into 6 different amino acids. All right, so fat digestion and absorption. So remember that fats have to be emulsified by those bile salts. And that's going to come from the pancreas and the small intestines. And when we break up fats, we're going to break them up into the glycerol backbone and those 3 fatty acid side chains. And the fatty acids and the glycerol are going to be absorbed by a process that involves several steps. And they're going to be absorbed into the blood or the lymphatic capillaries. If we're looking at the lymphatic capillaries, those are called lacteals. When we, let's see if it's on. When we digest and absorb lipids through the when we do fat digestion and absorption, you're going to need to know kind of these 5 steps. So we can kind of break them. We can need to break up these 4 steps into 5. So we're going to have let's just do this. So we're going to have emulsification by bile. emulsifi, emulsification by bile, or we'll say bile salts. And then we'll say hydrolysis by pancreatic lipase. All right, I wanted to kind of rework this slide. Okay, so fat digestion and absorption. So what's going to happen is we're going to emulsify these fats by bile or bile salts. And we talked a little bit about it up here in this slide. And one thing that I wanted to point out, because when I redid the slide that we were just on, I added something to this slide as well. So here, the only thing that I added was that these micelles, or they're basically complexes of fatty acids and bile salts that from which fatty acids can migrate to the microvilli and be absorbed. Because remember that hydrophobic and hydrophilic side. So these are the micelles. All right, which I talked about when we originally did that lecture before I paused and redid this slide. Okay, so we emulsified those fats and by the bile salts, and we did chemical digestion. We have the pancreas and the small intestine that are going to do its thing. We have that hydrolysis or the catabolic reaction by pancreatic lipase, and we take those fats and we break it down into that glycerol backbone and those typically 3 fatty acid side chains. And we can use those glycerol and fatty acid side chains, and we can resynthesize those into triglycerides in the smooth endoplasmic reticulum. Remember, the smooth endoplasmic reticulum is for lipid synthesis, whereas the rough endoplasmic reticulum is for protein synthesis. Anyway, those fatty acids and the glycerol backbone, they're going to be absorbed into the bloodstream by a process involving several steps. We're going to diffuse through the epithelial cell membrane, and then we're going to form these chylomicrons. And all they are just triglyceride clusters that are encased in these protein structures that are the micelle things, and we're going to move or absorb these micelles or the chylomicron things into these lacteal ducts, which go into the lymphatic system. Remember, the lacteals are like the lymphatic capillaries. So these chylomicrons are absorbed into the lymphatic system, into the lacteals, and then they are transported, or these dietary fats are transported to the muscle and adipose tissue, or adipose cells, through the blood, kind of packaged or emulsified. All right, so here we can see that the fat, this is our glycerol backbone here. These are our 3 fatty acid side chains. And then we have our water that's going to come in here and break up the 3 fatty acid side chains into the fatty acids and the glycerol. And remember that bile emulsifies these fatty acid side chains. And here's just kind of the process by what's going to happen. The fatty acid results from the triglyceride digestion entering the epithelial cells. Fatty acids are being used to synthesize triglycerides in the smooth endoplasmic reticulum. Triglycerides collected into clusters encased in proteins to form chylomicrons. Chylomicrons leave the epithelial cells, entering the lacteals. Then we have the lymph in the lacteals transporting the chylomicrons away from the intestines. All right, so here's just a summary slide of how everything is absorbed. We've talked about the monosaccharides, amino acids, fatty acids. We didn't talk about electrolytes and water, but we have in previous chapters. Electrolytes are move based on either simple diffusion or secondary active transport. Water is osmosis. Anytime we see water, osmosis is a diffusion of water. Movements of the small intestine, remember that still has peristalsis, that wave-like pushing movement. Segmentation is how the small intestine kind of breaks up that food into more movable or more usable chunks of food. So it takes and I'll just kind of reverse that order since we talked about mixing earlier on. We talked about mixing before we talked about moving. Remember that parasympathetic enhances the digestive system. So parasympathetic impulses are going to stimulate both the mixing and peristaltic movements, while sympathetic impulses are going to inhibit them. Now, if the small intestine, if you eat too much too quick, or if that small intestine does become over distended or irritated, you can see that's called a peristaltic rush. It's where a bunch of food enters into the small intestine too quickly. And basically your body's like, I don't know what to do. Let's go ahead and flush out everything and kind of reset itself. So this peristaltic rush is going to pretty much get rid of everything from the small intestine on. And so all of your food is going to go from the small intestine, peristalsis, small intestine, large intestine. You're going to have a bunch of diarrhea real quick. And then your body's going to kind of reset itself and say, okay, now I'm good. Let's go back to the mixing, and then peristalsis, and then hopefully that body has reset itself. Also, you might hear it called dumping syndrome. And then when we talk about the large intestine, or the connection between the ileum of the small intestine and the large intestine, we have this thing called an ileocecal sphincter right by the appendix that connects the small intestine to the cecum, and that's also going to help regulate the flow of chyme. So for the large intestine itself, it's only named large intestine because its diameter is larger than that of the small intestine. It's about 1.5 meters long. And then we know that the small intestine opens to the outside of the body through the anus. Basically, the purpose of the large intestine really is to basically absorb any ingested water, electrolytes, and to form and store feces, and to reabsorb any type of recycled water or digestive secretions. Now, remember that a lot of that your absorption happens in the jejunum. And so because the large intestine does not have any absorptive nutrient components to it, the large intestine is not going to contain microvilli. Remember, microvilli is there to increase the surface area for absorption. But because we're not actively absorbing nutrients from the food that we have ingested and or for chemical digestion, the large intestine is not going to have microvilli. So parts of the large intestine, we have the cecum, the colon, the rectum, and the anal canal. The cecum is basically this pouch-looking structure that is going to be the start of the large intestine. We can see the ileum is the last part of the small intestine, and it's going to empty into the large intestine. Here's your appendix. And there's your ileocecal sphincter, which is going to allow the ileum to dump its contents into the large intestine. Your appendix is there for immunity purposes. Then we have the colon. The colon is divided into 1, 2, 3, 4 portions. Your ascending colon, your transverse colon, your descending colon, and then we have this thing called a sigmoid colon. It kind of looks S-shaped a little bit, and then your rectum or your anal canal, your rectum. So on your colon, we have this we have these things called flexures. And basically it's just, instead of, your ascending colon and your descending, so we have like two kind of main curvatures of your colon, or bends of your colon. So we have a right bend and a left bend. So if we're actually looking at our body, this is actually the right side of your body, and this is the left side of your body. So this is your right colic flexure, and this is your left colic flexure. All right, so your rectum is going to extend from your sigmoid colon to your anal canal, and then your anus is just the opening of the your anal canal. There are two sphincters associated with your anal canal. We have an internal sphincter and an external sphincter. With your large intestine, we still have our 4 layers. The inside to outside, the mucosa, submucosa, muscularis, and serosa. Remember that the large intestine does not have any villi because we are not actively absorbing. It doesn't have this plicae circularis. Remember, we talked about plicae circularis in the small intestine for absorptive properties. But we do see that there's, we have longitudinal muscles that are organized into 3 bands that are going to run down the length of the colon. We do have these pouch things called haustra, which are going to help form feces. So what really does the large intestine do? It really has no digestive function at all. We don't do mechanical digestion. We're not breaking down big food particles into smaller food particles. We don't do chemical digestion. We're not chemically breaking down proteins into amino acids. We're not breaking down fats into glycerol and fatty acids. But we do have these goblet cells. Remember, anytime goblet cells are going to secrete mucus, and it says absorb ingested water, about 90% of the water, some electrolytes, but it has this intestinal flora, and that's just basically a bunch of bacteria that's going to break down contents like cellulose. Cellulose is just another name for like fiber. So if you are, let's say you eat a bunch of like plants and stuff, the undigestible portions of a plant are called cellulose, but in the plant world, but we call it fiber. All right. Also, the large intestine produces vitamins K, B12, and thiamine. Forms feces, carries out defecation. But it also has this, intestinal gas, which is, some nitrogen, oxygen, methane, carbon dioxide, hydrogen, ammonia, hydrogen sulfide. So hydrogen sulfide is actually what is responsible for the unpleasant odor in your farts. So there's your useless fun fact for the day. Hydrogen sulfide is what is the unpleasant odor in farts. So movements of the large intestine, very similar to that of the small intestine. We still have but we still have the mixing movements and the forward movement. So we still have mixing and peristalsis, but it's going to be slower and less frequent when compared to the small intestine. So, we don't, your if you eat a big meal, your small intestine is going to work for a long time after that big meal. But your large intestine, if you eat a lot, you don't sit on the toilet for a long period of time. I mean, unless you're a guy and you sit there and you play video games on your cell phone while you're pooping on the toilet. But, typically some people poop 2 or 3 times a day. Some people poop once every other day. But anyway, sometimes people, poop 2 to 3 times a day following meals. So we do have, the movement, the mass movement because of peristalsis. We do have a defecation reflex that you'll need to know, and that is how we eliminate feces from the body. And basically, if you want to force yourself to poop, you're going to hold your breath. Contract your abdominal muscles, lower your diaphragm, and that's going to force the feces to move into your rectum. And then peristalsis is going to take over and have that feces move into the descending colon, and then your internal anal sphincter and your external anal sphincter will relax, and you will see poop or fecal material, hopefully in the toilet and not your underpants. All right, so feces itself is about 75% water. And it just contains a lot of the stuff that we are not able to digest. Some electrolytes, the mucus, bacteria, bile pigments, and, not only do we have that hydrogen sulfide, most responsible for odor. So we can also look at, phenol it helps play a role in the odor. We already talked about hydrogen sulfide. Indole, scatol, ammonia is also going to be responsible for the unpleasant odor of your fart smell. Disorders of the large intestine, diverticulosis. Diverticulosis is basically a weakening of your intestinal wall, where your mucous membranes can protrude, and you'll see these outpouchings that can become inflamed or infected. You can see a little bit of the inflamed, infected areas here. We also have inflammatory bowel disease, abbreviated IBD. And we have 2 different diseases that fall under, or conditions that fall under IBD. One is ulcerative colitis, abbreviated UC, or Crohn's disease. Ulcerative colitis usually affects the mucosal layers and the submucosal layers of the large intestine. And you'll see, bloody diarrhea, some abdominal cramps. With Crohn's disease, that's actually going to be more serious. That's going to affect all of your layers, all 4 layers of your large intestine wall. It occurs in both small and large intestine. Again, diarrhea, pain. Here you can see the cobblestone effect within the tissues of the colon. So not only here you can see the redness inflamed, but you can see the cobblestone effect of the intestine. Obviously, one of the students or a couple students in one of my previous classes didn't know what cobblestones were, so I put in a picture of what cobblestones were, and then here's the picture of kind of like your intestinal wall and the cobblestone effect of that. Another thing, colorectal cancer. It's actually the fourth most common cancer in the United States. It's basically cancer of your large intestine or your rectum. We can do a colonoscopy. Now there's the at-home tests that you can send in a stool sample, and it's 92% accurate or something like that. Another disease or another condition that's not on here, hemorrhoids. Hemorrhoids is basically enlarged or engorged blood vessels of the anal canal or the anal columns. You can have internal hemorrhoids or external hemorrhoids. Let's see. Oh, loperamide. Loperamide is a medication that is used to slow or stop diarrhea. What it does is it inhibits nervous signaling to the large intestine. So it will relax the smooth muscle within the intestinal walls and prevent mass movements. So you would take loperamide for the conditions that have diarrheal symptoms. So, if you're looking at, ulcerative colitis, that has diarrheal symptoms. Crohn's disease has diarrheal symptoms. So, I think the test has a question about, it'll talk about loperamide, and it'll say, what diseases would you, or what conditions would you take loperamide for? And, you could say inflammatory bowel disease, or ulcerative colitis, or Crohn's disease, because all of those cause diarrhea. All right, lifespan changes. What happens? Your tooth enamel is going to thin. Your teeth may become more sensitive. Your gums can recede, so that means your teeth may loosen up a little bit. They may fall out. Your GI tract might become less efficient. You could have a thinning of your stomach lining. Your food can sit in your stomach a little bit longer, slowing the exit of chyme from your stomach. Peristalsis can slow down. So if peristalsis slows down in your small intestine, that means food will stay in your stomach a little bit longer. That can lead to heartburn, which means gastric secretions slow. Because food is staying in your stomach longer. Constipation may become more frequent. Nutrients absorption might decrease, including your fat-soluble vitamins. Remember fat-soluble vitamins, A, D, E, and K. Incidence of lactose intolerance increases. And then and that's about it. So that is it for this chapter. And so if you have any questions, please let me know. And I'll see you guys next time. Bye.